Welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. The views and opinions expressed by our guests are their own and do not necessarily reflect the views of the host, the American Phytopathological Society, or any affiliated entity. This is the first episode for 2026, and today I'm delighted to welcome Dr. Alejandro Olmedo-Velarde, Assistant Professor of Plant Virology in the Department of Plant Pathology, Entomology and Microbiology at Iowa State University in Ames, Iowa. Dr. Olmedo-Velarde holds a BS in Biotechnology Engineering from Universidad de las Fuerzas Armadas in Ecuador and an MS and PhD in Tropical Plant Pathology, Plant Virology from the University of Hawaii at Manoa. Dr. Olmedo-Velarde served as a postdoctoral research associate in Dr. Michelle Heck's USDA ARS lab, located on Cornell University's campus in Ithaca, New York, from 2022 to 2024, before joining the faculty at Iowa State University in 2024. Dr. Olmedo-Velarde's research program focuses on innovative management approaches for plant virus diseases and arthropod pests by dissecting and exploiting intricate multi-trophic interactions in vector-borne diseases in corn, soybean, grapevine, and other crops. He's published several dozen publications on diverse plant viruses and arthropod vectors and phytoplasmas. He currently teaches laboratory and microbial physiology, diversity, and genetics, and also serves as a guest lecturer for plant virology and methods in plant pathology. Dr. Olmedo-Velarde is an active member of APS, serving as current chair of the Virology Committee. He is also recipient of the 2025 Schroth Faces of the Future in Virology Award for his work on plant viruses and their arthropod vectors. Alejandro, thank you so much for joining us today.
Thank you so much for having me. I'm very excited to be here.
Currently, you're joining us from Iowa State University, where you are currently an assistant professor there. And what is your official title there?
Yeah, I just started last fall 2024. My official title is Assistant Professor in Host-Microbe Interactions. So my position actually it's a brand new one because it originated as the merge of the Department of Plant Pathology and Microbiology with Entomology. Two positions came, a colleague, Amber, who was hired before me, and then my position. So both of us have host-microbe interactions positions that try to merge plant pathogens, insects, and microbes.
So this is a new department that merges at least two historic departments. Is that right?
Yes.
Now, you're in Iowa now, but obviously you didn't start there. You grew up in Ecuador. Is that right?
Yes, I grew up in the tropics. Although I am not for the so much tropical weather, I am from the highlands. I am from Quito, Ecuador, and that is 2,800 meters over the sea level. That's pretty high. Just imagine having a forever spring weather.
Okay. And where you grew up in the highlands, was it pretty rural, or were you in a densely populated city, or?
No, it's definitely a densely populated city. I grew up in Quito, the capital of Ecuador. So right now, the whole metropolitan area is close to 2.5, 3 million of people. So pretty crowded.
Yeah. And while you were there, you got your BS and studied biotechnology engineering.
Yeah, so I obtained my bachelor in biotechnology in a university called Universidad de las Fuerzas Armadas, ESPE. It's another city called Sangolquí, and it's like a valley, so weather is definitely mild there. It's more like, even crops, you start seeing some little bit more tropical crops. So that's where I actually got all my background in biotechnology and microbiology, and particularly where my taste for viruses sparked.
That makes sense. I saw the engineering in the name, and I wondered if you were more an engineer-minded kind of scientist.
The university provides engineering degrees. So actually, I had to take all the math component as many other people following engineering degrees. But my degree, it's more like a blend of biological side, engineering side. So people say working in industrial side of production of microbes or things, or when they go to the environmental science part, they can definitely use more of the math part. But I'm glad I'm not using it so much.
I hear you there. Now, you did mention that you got an interest, or you caught a taste for viruses during that time. In what way? Could you give me a little more context there in terms of exposure to things like viruses during your time?
Yeah, the courses you take in my home country when you are pursuing a bachelor or even sometimes a master, it's very different to how you have it here in the US or other countries. So over there, honestly, you don't have much freedom to take the courses that you may want. Probably 95% of the courses, they are already pre-set up, right?
Okay.
One of the courses was virology as part of the reinforcement of your microbiology training and knowledge. So I took a virology course where I learned so much about viruses. At that time, I already loved all the microbe world, but taking virology, it was like, oh, really? This is like learning new molecular biology
Yeah.
because they are very unique in many aspects. And at that time, I just decided, okay, I want to be a virologist. And then, of course, I also took an immunology course. So I had to dissect mice and all that and do some experiments, which actually led me not to become an animal or human biologist because I was like, I cannot kill animals simply because of the sake of science. And then after that, got the opportunity to enter at Oklahoma State University that opened up my whole career through an internship.
Okay. The thing I like about virology is you get people in the door to a class like that, they're gonna quickly learn the importance of, like, tobacco mosaic virus, which was one of the first described and understood plant viruses, and in fact, its association with plants means that there's a little bit of plant science that comes into play in the history of virology.
Tobacco mosaic virus is the first virus ever characterized that we know it's a virus. So whenever teaching virology or talking about viruses, I have to say, yeah, plant virologists, thanks to plant virology, we know so much about viruses. But thanks to this opportunity that I entered at Oklahoma State University with Francisco Cho Corona, he's a virologist and biosecurity expert. I learned how grass studies work in the US because prior to that, I thought you had to pay or you have to be lucky to get into a reduced amount of scholarships. But after this internship opportunity, I learned so much about grass studies in the US, and I learned probably most people, at least in STEM, may not have to pay for your grass studies. So I really like that. And thanks to this opportunity, at Oklahoma State, I didn't stay long enough to attend an APS meeting, but then Francisco Choa took my poster into the APS meeting in 2016. Recall where it was held. I think it was somewhere in Texas or something like that, if I'm not mistaken. So that opened up the opportunity because my poster, I made the poster, Francisco printed it, took it to Texas, and then my grass studies PI, at that time, he saw my work on related viruses called Imara viruses. He was at that time working a little bit on newly described Imara virus he found in Hawaiian plants.
Mm.
So he thought there is a match, and then we started talking, and then that's how graduate studies came up. It was just actually thanks to APS on how, without me attending, I was back in Ecuador finishing all the paperwork and defending my thesis and doing some things, and then graduated. And as soon as I graduated, I went to Hawaii to pursue grad studies.
So that was with Michael Melzer, right? The PI who reached out to you about your poster and brought you on to study viruses in Hawaii.
Yes, it was Mike Meltzer. He's also another plant virologist, and he also knew somehow Francisco because they both of them work in the same citrus virus, citrus tristeza virus, which is pretty famous. And yeah, that's how everything, all my career started. And then during my grad studies, I was very fortunate to be able to interact not only with Mike Meltzer, who is a great virologist, but also Young Hu, who is actually Mike Meltzer's PI, but also he is the plant virologist. He recently retired and then recently hired somebody else. So I got the opportunity to interact directly with two plant virologists and get all my training and all the knowledge and learning many things like mentoring and management of projects and many things.
Oh, that's great. I'd like to go back to a point that you brought up, and it's worth mentioning again, is that your posters are seen at these meetings. Even if you're not there standing by your poster, if you put together a compelling poster, researchers, PIs, professors that are looking for recruiting students are seeing them, and this is evidence of that. And also that connection with APS, how it provides these opportunities to network with all the people you want to get your poster in front of. It provides a real opportunity. So that's great. Let's talk about your graduate work in Hawaii. And I do recognize that you completed 2 of your degrees there, a master's and a PhD. So a lot of time there, a lot of work. I mentioned previously in our email correspondence that I was really impressed by how many diverse viruses across so many diverse plant hosts that you worked on, from hibiscus and orchids, passion fruit, taro, fig. It probably speaks to the botanical diversity of Hawaii.
Indeed.
At Cornell and now in Iowa, like, I feel like you work on fewer viruses and phytoplasma diseases on agronomic crops. But like, how do you think that work in Hawaii prepared you for like where you are now? And do you want to talk a little bit about some of the more interesting work you did in Hawaii?
Yeah, my time in Hawaii was very rewarding, very enjoyable because agriculture in Hawaii is very diverse. Of course, there is the top commodities. I think seed corn is still one of the main commodity in the island that moves so much economical input. But then because of probably the history of the islands and my Asian and American migration, because Hawaii is right in the middle of Asia and America. So that creates a unique scenario for agricultural practices and agricultural history. So you get all these crops being grown, especially many of these Asiatic vegetables, that it was my first time seeing them. And the diversity of crops is just amazing. And the fact that Hawaii is also in between Asia and America, that also creates a door for moving pests and pathogens. Living in Hawaii was a kind of a dream for characterizing and seeing new stuff because they were coming and showing up because of trade, because of globalization. It's sometimes just simply impossible to control how much what you are moving into and out. Well, we move things, we trade, and that's just part of our economy, but that's also how we move plant pathogens in general. And that was also part of the unique opportunity I got being part of agrosecurity laboratory because Mike Meltzer, even though he's plant virologist, he's doing agrosecurity research in general. So that also exposed me to not only plant pathology research, but also entomology research. And soon enough, I learned if you want to work with plant viruses, you got to work with their vectors, right? Most plant viruses have biological vectors, which is arthropods. I was slowly being forced to work with the vectors. That was a great opportunity and very enjoyable opportunity for me. And because of that, we would receive plant samples from vegetables, some other specialty crops, and so it was just great.
Yeah, absolutely. And also getting into working with arthropod vectors, that kind of like then position you to move to Cornell, where you worked in Michelle Heck's lab, and they do a lot of important work with vectors there, which I assume you just further developed your skills in working at that interface between vector virus interactions, the 3-way interactions, virus, plant, vector interactions, right?
Yeah, during my grad studies, I indeed really got so many biology skills, knowledge, and also this vector biology knowledge indirectly, right? Working with a variety of vectors, because again, the variety of viruses that show up, you start learning, oh, this is the potential vector. It's not only just aphids or few, it's a variety of things that we were exposed to start working. And then this opportunity just led to, at that time, X and Twitter at least it started emerging more, at least in my site. So I created an account. And then among the people I was interested in potentially doing a postdoc position, there was Michelle Heck. So fortunately, it was a match because she had the funding for a postdoc and I was just about to graduate. So it was just about time because sometimes even there are many great candidates, but sometimes the funding is not there. So the timing was just perfect. And I was able to join her laboratory. And I have to say this briefly, it was not only me. I am married to another plant pathologist. She's also actually now working at Iowa State University. And she's not only a virologist, but also a bacteriologist by training. even though sometimes we try not to take science and the work-related topics to home, is sometimes just impossible, right?
Yeah. During your time in Hawaii, just to kind of rewind a little bit, what was the most interesting virus a pathosystem or virus plant interaction that you worked with?
That's a great question. So during my grad studies, my Mike Meltzer, he's an expert on many viruses, but one of the viruses he likes the most probably are called Brevipulpus transmitted viruses. Brevipulpus is a genus of flat mites. So they are micrometric. And at that time, it was a huge learning curve for me to learn to be spending time under the microscope, trying to learn how they behave and all that. And they are micrometric mites. Those were one of the most interesting plant viruses because when a plant virus comes and infect a plant, they normally infect the whole thing, infect the whole plant from the leaf that they enter through, and they even go to the roots, right? So different from most plant viruses, these Brevipulpus transmitted viruses, they cannot infect the whole plant. Instead, they stay locked in these localized lesions that even though they move from cell to cell, they cannot reach vascular compartments. And this is the hypothesis. So that also expanded my interest and expertise in developing a virus infectious clone for this type of viruses, because I thought, okay, this is a skill I want to learn. Nobody else has developed such tool for these kind of viruses. So that also expanded my interest and my expertise in doing biology work. So indeed, those are the most interesting plant viruses, because even though they stay in this localized lesion, there are this disease called citrus leprosis that is caused by a variety of viruses. It's very important in Brazil. We talk about citrus greening, then it's definitely worldwide important. But talking about Brazil, you have citrus greening and they also have citrus leprosis. So a number of people there working in these kind of problems in different aspects. And then Hawaii is unique. It's not part of what we call the mainland, right? 48 states. And so we had those viruses, but they are not present in US mainland. They are just present in Hawaii.
Yeah, I see the challenges of working with something like mites because they're obviously an interesting group of arthropods, but they require a whole different level of skill than maybe even not to downplay the diversity of hoppers or other insects in their roles, but mites can be challenging, especially something like a flat mite. So I want to talk a little bit about the fact that you went from really being very virus centric, maybe viroids, to working with like other fastidious prokaryotes, phytoplasmas and whatnot. When did you start working with those? And could you contrast them to working with like viruses and that, and like which ones present more challenges in working with? Because they both have their own issues in terms of how well you can work with them at the bench. So
Yeah, that's a great question. When I just started my plant pathology training in Hawaii, I learned about these non-culturable plant pathogenic bacteria, right? And seeing how they behave and everything, I thought, okay, they are just like viruses to some extent, like in the practical sense, because you cannot culture them. You want to maintain them. You have to either use their vectors or perhaps grafting or vegetatively propagated the crops, right? That led me already to being interested in how learning more, and also again being part of an agrosecurity laboratory. At that time, my laboratory was also doing surveys for citrus greening, because Hawaii is very unique in the fact that has the vector, but so far doesn't have the disease. Citrus production is not like it used to be before, but it probably has ups and downs, I would say. And then because of that, definitely citrus greening is one of the most important things that you want to learn if it arrives, because the vector is already there. So because of that, learning that the molecular work is required. And then when I came here, it was to Iowa. I already knew about cornerstone, and it was like a very interesting disease. And I was thinking how we have so much to still understand and study compared to other crops. And I thought, even though that's a so interesting disease, because it's limited to the tropics, it's going to take me a few years to be able to work permits, then get the isolates and all that. But then as soon as I arrived, then I learned, okay, the vector is moving northwards. So there is a chance it's also here. And it was true. Surprisingly, the cornleaf hopper was already in Iowa when I came.
Uh-huh.
And the disease it was also here, although to be being very brief, impact at least in the state has not been as at least that year we did not see so much impact. But the disease also came, and it's all about timing and many things that we can expand later.
And the disease you're talking about, is this the maize bushy stunt?
You can call it maize bush stunt or corn stunt. So most literature called corn stunt disease that it's caused by either corn stunt spiroplasma or maize bush stunt phytoplasma. Different from actually corn stunt spiroplasma is culturable.
Okay.
There is not easy, but some people have been able to culture different from phytoplasmas. We have not been able, nobody has been able to culture phytoplasmas yet. And that the disease is caused by either pathogen, because the vector is the cornleaf hopper, it is what is moving the pathogens of the disease. And the symptoms are pretty similar. So that's why this is just called corn stunt disease.
And just to clarify, can you get co-infections of both the phytoplasma and the spiroplasma in the same plant?
Yes, you can get co-infections, and that's what it has been reported for so many years, particularly in Brazil and nowadays northern Argentina, but also nowadays in the southern US, you say Texas, Oklahoma. Actually, the disease and corn is native to Mexico and the cornleaf hopper as well. So the disease was just in Mexico for so long. And then the actual disease was first characterized in the Rio Grande Valley in Texas. So it was just a matter of time that it came to the Midwest, to the Corn Belt. And let's not be surprised if it was not the first time that it actually came. Perhaps it was just the first time that we noticed.
Yeah, and like just reading your work, it's clear that data mining can really help us clarify the origin of a lot of newly established pathogens. And you've published some work recently where you do that using, you're mining old data sets and molecular data and able to actually show that no, things were here a decade earlier. It's just that you have to come up with creative ways to look through existing data sets and create a more comprehensive snapshot of what's currently out there, right?
Yeah, data mining it's a broad term that is pretty much you reuse what's already publicly available, right? And particularly this, say, practice really emerged during COVID because there were a number of researchers and particularly virologists sitting in their home because of COVID. And many of them are starting, okay, we cannot generate data sets during this time, but there are so many data sets that can be reanalyzed. And particularly at that time, soon after that, then there is this virology data mining database that is called Ceratus that you can explore, and they actually already did the work for you. So you can just explore the database and you just type the virus you're looking for, and that's already a great tool to do it without you having to have the computer power to explore by yourself. But of course, limitation of this database it's dated up to 2020. It's been 6 years, so probably it's time for an update. It was during my postdoc time. I was working with this infamous virus, cotton leaf rollworm virus. So it was first found in the U.S. in, I think it was Alabama or Georgia. It was somewhere in there. And then it was associated to severe yield losses, and particularly that has been also reported in Brazil and Argentina. So we just shared so much agricultural things and diseases with the southern counterpart.
Yeah.
And particularly also seed companies, they both for row crops, they work in US and Brazil and Argentina, right? There was this virus that was associated to severe yield losses. So influx of funding started coming. Michelle Hegg is an expert on this type of viruses called polioviruses and luteoviruses. So she was assigned to work with this kind of virus, even though her laboratory is still at Cornell University. She was assigned to work, and this because of the unit, the ARS unit that is working with emerging diseases and pests.
Nice.
One of the main thing is that I was wondering, it's like, even though the virus was associated to severe yield losses, when many other people started testing plants, including asymptomatic plants, they would find that even though the virus would be there, may not always cause symptoms. This was not a new thing for virologists because there are a complex of factors like when the disease or the virus arrived. Perhaps the plants were already too old. Even though they get infected, they do not show much symptoms. Talk about cultivars. Just the number of cultivars that we have for our row crops in the US per stage is just tremendous, right? So many of them. So there are, and plus environmental factors, right? But still, I was probably there are other viruses, right? Or perhaps there is a chance that this virus was already here and then something happened. And then there is this kind of things that when the virus is already in the country for some time, and then as you develop cultivars, they learn to interact with the virus. And that's why not so much symptoms show up, because also breeders look for the healthy looking plants. So that's actually you are artificially selecting perhaps without knowing. And that could be perhaps some scenarios. By using data mining, I found that evidence. And one of the most important evidence I found, it was through this very old database that actually before trying it, I didn't know the existence of it, and the database is called Express Sequence Tags. So by checking what it is, just some years or decades ago, we were trying to figure it out. We have DNA sequences, but we were not great at predicting where the genes are, right, in the genome. So at these express sequence tags were just struck RNA, make cDNA libraries, clone it, and start sequencing those clones to start mapping where those genes are. One of these bunch of clones were generated at that time from a virus-infected plant. But my guess is that the researchers didn't know perhaps it was a virus-infected plant. And they uploaded this bunch of clones. And of course, one of these sequences that were uploaded to GenBank were actually of virus origin of cotton leaf rolled virus. And by doing the phylogenetic analysis, it pointed out to be pretty related to what we already know. It is already here in the US. So further expanding the hypothesis that the virus was already here. We just didn't know it. And at this point, if you ask me, there is no evidence that the virus was introduced, because some people say, oh, perhaps it was introduced from Argentina, Brazil. We have done the studies and we have sequenced so many genomes. All of them, they have their own cluster. So there is no evidence for us so far to tell the virus was introduced. And this data mining approach and study just told us the virus was not only here long ago, close to 20 years ago, but also it is has been here and perhaps evolving by itself.
I think it's pretty common for people to jump to a introduced pathogen hypothesis when something emerges seemingly suddenly on the landscape. And it's good that, there are people doing the work retrospectively and looking at this data. One of the other things that have come from data mining, and we think about all these metagenomes that are being sequenced and published. We also have RNA-seq data sets, DNA sequencing. I think you've been involved in publishing some genomes that were sequenced alongside something else. We've done this with bacteria. You'll get whole bacterial genomes in a fungal genome because they were co-occurring in the sample. And that's true of viruses too, where you can basically assemble these virus genomes inadvertently. There's a lot of people that deliberately now want to sequence the metagenome there and extract out the virus sequences, but certainly there's a lot still to be discovered in existing data sets, right?
Yeah, so this also comes back to the fact what I was just mentioning about cotton leaf rolled virus present also in asymptomatic as well as symptomatic plants, right? So I thought it is also possible that the virus it's not alone. There are other viruses there. So one way to do it is metagenomic, metatranscriptomic studies, right? Using the power of omics. And through these studies, that's what we found. Initially, there were insights that were not clear because the genome organization was not typically looked weird. And then after some thinking, also I recall, okay, check the family. This virus is potentially belongs to this family of viruses. And then these family of viruses have been reported to be integrated into the genome of their host.
Yeah.
So that's a feature that has been already reported and has been characterized. Some people say well characterized, some others say not so well characterized. But if you ask me, characterized for a banana virus, and that it is particularly important when these viruses are integrated into hybrids. And this has been when we grow bananas, they are hybrids, right? And they have A and B genomes. And this banana virus has been reported in the B genome. Checking the history of how we grow cotton, it is allotetraploid. So long, very long ago, it was just two different species. And then nowadays we can actually produce true seeds that are viable. But very long ago, it was two different species that they had the A and the D genome. And this virus was found integrated mostly in one chromosome in the A genome. It was a very interesting finding, and also all the work was possible and expedited because of the high quality chromosome scale genome sequences that we have for cotton for many cultivars.
Yeah.
And also the computer power that we have, the tools. And it was some work that took some time to figure it out. We were not only the group that found this, it was also another group in Mississippi. And that just reinforced the fact that the same thing that you find, another group also found that it's there. And of course, it's uncertain if the virus, this new virus, is really causing anything. It was probably the most challenging virus I had worked with because the fact that it's integrated into the genome.
Sure.
We were never able to get it in high concentrations or anything like that. Or even the fact that it's integrated into the genome, we were not able to develop a PCR assay that can clearly distinguish the integrated part from the virus assay that is high throughput, right? It was through virus purification that we were able, okay, we indeed have a virus. Plus, not only the integrated part. It was one of the most challenging and rewarding experience at the same time.
All right. Thank you for that. We're going to shift gears a little bit here. You're part of the Iowa Soybean Research Center, and we've talked a little bit about maize and cotton viruses. Tell us more about your role with soybean research in Iowa, and tell us, are there a lot of viruses, those kinds of problems that are impacting soybean there compared to other plant pathogens like nematodes and fungi, things like that?
Yeah, when I just came, I thought, okay, normally you want to focus on what you have in your surroundings in the state and make local people happy, right? So definitely I had to focus on corn and soybean viruses or vector-borne pathogens. In the case of soybean, indeed, viruses can be a problem. But when you think about soybean cyst nematode or soybean sudden death syndrome, it's not like that, right? So it's simply the prevalence is not too much. And also it depends on, again, environmental conditions, cultivars, and also the presence of biological vectors. If you ask me, when I was just driving from upstate New York to here to Iowa, and as soon as I entered the Corn Belt, it was just amazing to see how well corn and soybean do, like very uniformly as you drive. Yeah, I was super impressed. And that's also the great cultivars that we have, even though they are still susceptible to some things, at least to viruses, they are not so much as you may find other countries are dealing with virus problems, but still viruses are there. And the Iowa Soybean Research Center provides a platform for researchers in Iowa doing any kind of soybean research, a platform to network, collaborate, and also learn about the soybean research that's going on. Very importantly, also provides an opportunity for internal funding that it's allocated yearly. It's pretty competitive. Actually, one of my ideas using virus structural proteins and toxins to control soybean aphid populations was a finalist, but unfortunately was not selected for funding. But I found it as a very rewarding experience being a junior and new researcher here in the Iowa State community, that my ideas were recognized.
Sure.
Yeah, so I've been 1.5 years in my laboratory, still building my research program, but my laboratory members are definite. I feel very fortunate also that I have a great startup that has allowed me to develop my ideas and develop my team and then start applying for competitive grants. And also, again, the Iowa Soybean Research Center facilitates that through these internal funding opportunities.
Sure. And I saw that on your CV. You have a lot of pending funding. So it's clear you're putting in a lot and looking for a lot of opportunities in the future. You're the 2025 recipient of APS's Troth Faces of Future in Virology Award. Tell us more about that and what that award means to you.
It means a lot. I feel very honored and humbled when I learned I was selected for the award. And this is also date back to when I was just starting in the plant virology world with Francisco Choa and with Mike Meltzer, because I learned more about APS and everything, and I learned about this award. And I don't recall exactly which year because it rotates the area, right?
Yeah.
Every 4 or 5 years goes back to, so in 4 or 5 more years, there will be another virology recognition. At that time, I saw it's amazing that APS has this kind of recognition that you feel valued about your contribution as a virologist, as a junior researcher. And I thought since then, I hope I am able to at least apply and hopefully I can get it. And again, I feel very honored that I was selected because I have been fortunate that I had been, is not only thanks to my effort, but also to the opportunities and to the great support that all the people and mentors I have had during my grad studies, my postdoc. It was a great thing to receive.
Yeah. And I interviewed recently Tiffany Low Power, who was a recipient of that for bacteriology. And she spoke similarly about it feeling like a real honor and inspiring you to keep doing what you're doing. It's nice to be recognized that you're helping to advance a field or a subfield. Certainly that you are. One of the interesting things on your CV that caught my attention was your 2024 phytopathology paper. You were establishing a reverse genetics tool that allowed you to fulfill Koch's postulates and hibiscus green spot virus 2, if I got that right. As someone who teaches general plant pathology, we spend a lot of time talking about, the caveats and the barriers to fulfilling Koch's postulates. And when you work with obligate biotrophs or viruses, or there's a lot of room for problems in trying to kind of fulfill Koch's postulates. Will you talk a little bit more about what you accomplished in that paper and how that allowed you to kind of fulfill Koch's postulates in this virus system?
Yeah, hibiscus green spot virus 2, it is what I told you, what I mentioned previously, the citrus leprosis virus, although some people like to call it citrus leprosis because indeed doesn't have or does not cause all the symptoms that citrus leprosis viruses cause. But at that time, it was a virus that was unique. Number one thing, because Mike Meltzer characterized the virus, so you want to make your PI happy doing an infectious clone for the virus that your PI characterized, right?
Sure.
And also because at that time, the virus was only present in Hawaii. It was found nowhere else. It was just present in Hawaii. And it was in the backyard of the building where I was doing grad studies. And because it's Hawaii, right? You don't have winter break, you don't have all these temperatures that kill insect or arthropod populations. So
Sure.
arthropod populations in Hawaii have natural ups and downs, but they are always there, which means also you can get the virus all the time. And because of that, I thought, okay, this is a great opportunity for me. Talking about Koch's postulates, right? And a virus infectious clone. Virus infectious clone is just a way to deliver the genome of the virus to the host. And that is going to start virus infection. So in pretty simple terms, and there are different ways one can do it. Of course, when we talk about plants, we have to rely on one of our best friends and natural genetic engineer, agrobacterium.
Sure.
So through agrobacterium delivery, we deliver a plasmid harboring the copy of the genome of the virus with the adequate promoters and signals so that the transiently expressing cells will actually produce the virus genome, and then eventually that's going to start virus infection. For plant biologists, that's one of the few ways that we have to fulfill Koch's postulates because, say, animal and human virologists, even though viruses are not culturable, right? But animal and human virologists, they have cell lines that can sustain virus infection, and those cell lines are culturable. And say COVID, actually, you can culture COVID in vivo and some other cell lines. So we don't have that for plant viruses. And because of that, one of the ways is to use a virus infectious clone. And sometimes, if you perform properly, is using the vector. But coming back to the virus infectious clone, you are delivering the virus through the infectious clone to the healthy host, and then should develop disease or at least symptoms. You should be able to retrieve the same kind of virus, right? The same genome or having the same properties. So the development of this tool was fundamental for Mike Meltzer Laboratory to continuing a research line on this kind of viruses. Currently, a PhD student is using this tool to further the studies of this particular virus. And at that time, again, help us to definitely corroborate that this virus is causing the symptoms that we were seeing in the field. Because again, another way was using the vector, but using mites, micrometric mites is not easy at all because just establishing colonies, it's very challenging and complicated. And
Yeah.
I do prefer working with aphids or any other arthropods.
Sure. And just to remind our listener, this was earlier when you were talking about this virus, you said this is not like a lot of other viruses in that it doesn't lead to systemic infections. It stays localized even as an infectious clone that's being delivered through an agrobacterium plasmid, right?
Yeah, so it just stays mostly where you deliver agrobacterium, could move to few neighboring cells. And this is a kind of plant biology knowledge gap that we have for these viruses because it's not that these viruses do not have, because all plant viruses need a movement protein or group of movement proteins that help them move from cell to cell because of the cell wall, right?
Sure.
They can do that, they can move from cell to cell, but it's simply they cannot reach vascular compartments and move systemically. There is a chance they take way too long, and then the plant trigger a hypersensitive response. So that's already has been determined for one of these viruses, and that's also, if you think about if it triggers hypersensitive response, this is why citrus leprosis virus or citrus leprosis disease has the name for, because when you have great populations, many feeding sites, and many necrotic sites.
Yeah.
That's what citrus leprosis is about. And in the case of this citrus leprosis virus in Hawaii, doesn't cause so much necrotic symptoms, but it's still here and there. And that was one of the main things at that time that at least helped to develop a tool that we have now to better understand these kind of viruses.
So does that mean that because you're not necessarily seeing a hypersensitive response, there could be other quirks to this, for lack of a better term, to this virus that could give you unique insight into the larger discipline of virology because there's something about it that makes it unable to reach that vascular tissue, even in the absence of an outward hypersensitive response.
Yeah, and even some when you check the phylogeny and like these viruses are here, and when you check the phylogenetic tree of related viruses, not surprisingly, there are viruses that have been found infecting or associated to arthropods only. So if you ask some virologists and even perhaps Mike Meltzer, also I do agree, perhaps these are not so much plant viruses. These were arthropod viruses that learn to infect plants, but they are still learning. And this is why the plant can still sustain or control the movement. But again, this is just a hypothesis, right? To have this hypothesis, there is the phylogenetic evidence and how they share some genes with arthropod-specific viruses. But because these viruses do not infect very, say, raw crops or other commodities that can be a little bit more well-funded, there is still so much to be done.
Sure, that makes sense. Believe it or not, we've pretty much exhausted our time together here, and I've really enjoyed talking with you. Thank you so much for your time.
No, thank you so much for having me, Matt. I'm very happy that I had the opportunity to talk with you about all these chronological things I have done during my career, and I'm excited to see what's next for my program because I am somebody who likes to adapt, and as you can see, I'm not only stayed with plant viruses, I adapt to what is needed for the region. In this case, I started working with phytoplasmas, spiroplasmas, and leafhoppers. So I don't know where my program is going to be in 5 to 10 years, but of course, working with something that is relevant and needed for the area and for the U.S. economy.
We've just heard from Dr. Alejandro Olmedo-Velarde, Assistant Professor of Plant Virology in the Department of Plant Pathology, Entomology and Microbiology at Iowa State University in Ames, Iowa. As with our previous episodes, be sure to check out the show notes immediately following the episode description for links to Dr. Olmedo-Velarde's research program. I'm Matt Kasson, host of Plantopia. Thanks for listening.